Hello students, we are starting off with the second series of developmental biology, that is unit 5 in CSIR net life science portion. This will also be helpful for icmr people, CSIR people and other life science competitive exam students. We will see about what is developmental biology, what a candidate will study under developmental biology. In developmental biology is about the study in which, in basic terms, in practical terms, it is a term in which it is study branch of science that deals with the study of how in case of plants, how the embryo will develop into a whole plant, okay, how the fetus is formed, how the fetus will progress in its stages, how it will become a complete baby, developmental stage of fetus, developmental stage of plants in amphibians, how the undifferentiated mass of that is stem cell can differentiate into so many other type of cells. These things will be dealt in this thing. With regard to developmental biology, we are supposed to understand basic terminologies like what is growth. What is growth? For an embryo to develop into a whole plant, it should grow, right? You should understand what is growth. What is differentiation? What is growth? Divide, differentiate, pattern, it is specification, morphogenesis. In order. First growth, then divide, then differentiate, then pattern formation, morphogenesis. Growth. A cell has to grow. Cell has to keep its alive first of all to grow. Cell has to divide. Cell has to different. This cell has to become heart cell. Cell has to specify, and it should be determined to become a particular cell of a body. So it is differentiated. An undifferentiated simple cell is differentiated to become specified and determined to become heart cell, nerve cell, skeletal cell, muscle cell, whatever. So this is growth and differentiation and division. Cell should multiply, cell should grow, cell should divide, cell should kidney cell, heart cell, muscle cell, nerve cell, cell undergo differentiation. Pattern, organization. Pattern in the sense organization. They are organized under every system. Shape and structure. A cell cannot wander alone inside the body. It needs a system. It needs coherence. It needs circulation. It needs nourishment. It needs oxygen. It needs a shape. It needs a structure. Okay. First up, what is stem cell? What is stem cell? Stem cells are the undifferentiated Stem cells are the undifferentiated mass of the cell. Cells are in case of an in vitro medium, let us consider an in vitro medium. We have a culture plate, we have so many type of cells inside it. These are undifferentiated, but these cells have a potential to develop into any kind of cell. So far it is not differentiated into, and it is not determined to become any particular type of cell. These cells are called as undifferentiated cells. They are called as stem cell. Okay. What is stem cell? Niche. Stem cell niche. Consider a culture medium. Inside the culture medium, we have stem cells inside the medium. Okay. It is said that a cell will release a paracrine factors to other cell. The cell will respond to it, other cell. These paracrine factors, these green colors are known as paracrine factors. This paracrine factor will maintain the stemness, that is undifferentiated condition will be maintained. Undifferentiated will be maintained by these paracrine factors. Paracrine, so these cells are not committed. So the stem cell, they are not committed and determined to become any particular type of cell. This nature is being maintained by these paracrine factors. This environment is called a stem cell niche. Okay. Now we will see about potency. What is potency? Basic terminology with regard to developmental biology. What is potency? An undifferentiated cell has a potential to differentiate into any type of cell in a body. Okay. Consider a factor. We have a culture medium. We have an undifferentiated cell in it. We will study this undifferentiated cell now. This undifferentiated cell has a potential to become heart cell. If it is induced, it can become heart cell. It has also a fate to achieve nerve cell, muscle cell. It has fate to become kidney, whatever. If it is receive the signal, appropriate induction signal. Okay. So this undifferentiated cell has very high potency. This is what with regard to potency. Ability of a stem cell, that is undifferentiated, to form many different type of cell, etc. To form many different type of cells. Talk about basic terminologies with regard to potency. Totipotency. First is totipotency, pluripotency, multipotency, unipotency. Okay, we will see about each thing. Basic details. Without these, we will not be able to understand the whole unit. Totipotency is embryonic stem cell. Pluripotency is also called as embryonic stem cell. Cell has a potential to develop into whole organism. is called as totipotency. In pluripotency, a cell has the ability, is potent to become whole cell except trophoblast. Except trophoblast. So what are these trophoblasts? When the cell is produced, the outer layer is primarily formed. These are called as trophoblasts. This is called as pluripotency. Multipotent, you will see about multipotent stem cell. What is multipotency? In multipotency, a cell is committed to a small extent. For example, consider hematopoietic stem cell. Consider hematopoietic stem cell. It has a fate to give rise to other blood cells. It can give rise to RBC, it can give rise to WBC, it can give rise to platelets, it can give rise to only blood cells. This cannot give rise to heart cell. This is called as multipotency. Okay. Next, about unipotency. What is unipotency? Found in only particular tissue. It is capable of regeneration. It is capable of regeneration. Example is stem cell, that is spermatogonia. Spermatogonia. It will give rise to only sperms. It will never ever give rise to other cell. This is one such example of unipotency. We'll erase this and we will make it clear. Unipotency, capable of regeneration. And it will give rise to only particular kind of tissues. Example, oogonia, spermatogonia. Give rise to ova. It give rise to sperms. Example of unipotency. I hope in this video we have cleared about basic definitions of developmental biology, that is stem cell niche, potency, we have seen about various type of potency, totipotent, pluripotent, totipotency, pluripotency, multipotency, unipotency. I hope so. All these terminologies are very clear. Understanding these terminologies are very important in moving further to developmental biology series in this video. Only then we get to understand about development of chick, development of C. elegans, dorsal ventral axis patterning, Drosophila. We will see all these in detail only if we get to understand the basic terminologies. with regard to developmental biology. Hope this video is very clear to everyone who prepare for this exam. All the very best to everyone from CSIR point of view and also for other life sciences examination. Best wishes to all candidates. Thank you.